Hello, and how are you? Welcome to my session on regulation of cell life cycle. My name is Winnie Barawa. If you're new here, this is a channel that does lectures and lessons pertaining to healthcare. Please don't forget to subscribe and keep with me all the way to the end. So in the previous lecture, we did a description on what cell life cycle is, but we did not explain how do we regulate the cell life cycle stages. I want you to note that the cell life cycle is regulated and there are different mechanisms of how the cycle is regulated. So this is the main focus for this class. And if you've not watched the class on cell life cycle stages, go ahead and click on the link and be able to be updated on the same. So in the cell life cycle, we say that the cell goes through four main stages, starting with the gap1 phase, S phase, gap2 phase or G2 phase, and the M phase or the mitosis phase that contains mitosis and cytokinases. But now today we want to see how do we ensure that this cell is not damaged or we don't not end up producing damaged cells or cells that don't have the proper So there are different mechanisms of regulating the cell life cycle, and one of these mechanisms is the presence of checkpoints or what we call restriction points, R. So what these points do, they detect if a cell contains damaged DNA materials and ensure that these cells do not replicate and divide further. So these points are located at different areas of this life cycle phases or stages. For example, we have a restriction point located at the G1 phase, and this is a very key checkpoint. Reason being, majority of these cells that pass at this point will be checked if they have the proper DNA content, they have the proper organelles, or that they have the proper structures that will be required to ensure that they go through the whole cycle process. So the checkpoint that is present here will eliminate this cell provided that they have detected there are some damages and there are some qualities that are not worthy being replicated or being divided at the end of the cycle. So most cells that are not proper get eliminated at this stage. But where again do we find these checkpoints? Other checkpoints are located between G1 and S phase, and then we have another checkpoint that is located between G2 and the M phase. So let me use a diagram to make this further clearer. So here are our checkpoint. We said the most important checkpoint is at the G phase, that is G1 phase or the growth phase. So at the gap1 phase or the growth point check, we have this checkpoint here. So at this checkpoint, we want to check the status of the cell before it goes further to the next stages. So at the G1S phase, we have a checkpoint there that looks at the environment. That is the environment proper enough for the cell to go through the cell division, or there's some characteristics that are not friendly or they may hinder the growth and division of the cell. Then the other bit is looking at the size and shape and requirements of this cell. For example, is the cell large enough for it to divide as it goes ahead? And the other bit is the DNA quality or the DNA quantity. Is it in its required amount? Is it in required components? Or there's some damaged DNA materials that will cause some mutation and damage this cell? So once this has been confirmed, then this cell can progress further and go to the next phases. So we have in between G1 and the S phase. So this point or this checkpoint checks for three things: environment, the cell size, and the DNA qualities or DNA characteristics. And the other checkpoint that we're mentioning, we are saying that there's a checkpoint that is found between G2 and the M phase, or the G2 and the G2M checkpoint. So the G2M checkpoint, what it does, you find it between the transition from G2 phase to the mitotic phase. And what this checkpoint will do, it will check to confirm that the DNA has fully been replicated, or it is complete and contains each and everything that is needed. And then it ensures that the environment is also ready for the cell to transit to be divided. So once at G2 you've confirmed the DNA has fully replicated, it's fully formed, the environment is proper, then the cell can now transition from G2 and begin the cell division process. And once the cell division is ongoing, we are also being told that there's a metaphase checkpoint. There's a checkpoint where we check the status of the metaphase. When we discuss mitosis, you will realize that at metaphase stage, this is the stage whereby the spindle fibers will be attaching to the chromosomes and allowing it to move to the ends of our poles. But we'll come back to that later. But for now, let's appreciate that there's a checkpoint that is found at the metaphase during the mitotic phase. And from there, the cycle goes in. So basically what we are saying is that our cell life cycle has a regulation mechanism that uses the checkpoints or the restriction points. Famously, we shorten it as R. Second mechanism that helps us to regulate our cell life cycle is the use of the p53 proteins. The p53 protein says that if a damaged DNA has been detected at any of these checkpoints, remember we are saying the first mechanism is a checkpoint. So if in this checkpoint you detect a damaged DNA, we are told that the presence of this damaged DNA will activate production of p53 proteins. What these p53 proteins are, they are tumor suppressor genes that stop the progress of this cell from going to the next mechanism. So anytime we produce a DNA that has some damages and it has some potential to cause harm in the future stages, what happens is the p53 proteins are produced. And what these p53 proteins will do, it will arrest the process of cell cycle, or it will stop this process from happening, and then it will ensure that the damaged DNA has been repaired. So it's like a break. So when you discover that at the checkpoint you have seen some cells that have damaged DNAs, the best thing to do is stop the process. So what stops this process is the p53 proteins. So the p53 will stop the cell cycle, and then it will allow time for the DNA to be repaired and be made to be complete. If the DNA fails to repair, what happens is we will now kill the cell, or we will let the cell undergo a process we call apoptosis. So apoptosis in definition means programmed cell death, or rather what we are saying is we will allow the cell to die or to undergo death, and it will no longer go further with the replication. So this is what happens. When we are going through the checkpoints at the G1 S phase, we will maybe find out a few of these cells have damaged DNA. Then what we'll do is we will have a release of p53 proteins. So what this p53 protein will do, it will arrest the cell cycle. It will not allow the cell cycle to go on. So what happened first, we will repair this DNA. If the DNA gets repaired and complete, then you will restart the cell cycle again, and the cell will go further, complete its process, divides, and go on with the cycle. But now if the DNA fails to replicate, or the DNA fails to repair properly and allow its structures to be complete and normal, then the cell will go through apoptosis. What we are saying, programmed cell death. So the programmed cell death, what it will do, it will eliminate the damaged cells that have the DNA that is incomplete or is damaged. And with that, we'll only allow the cells that are complete, have proper DNA qualities, they go ahead with the process. But the ones that have damaged DNA, they'll simply die and be eliminated from the process. And the third regulation process is the use of cyclins. So what cyclins are, this is a class of proteins that control cell progression by activating what we call cyclin-dependent kinase enzymes. Cyclin-dependent kinase enzymes, mostly known as CDKs. So what cyclin do, they will be binding to these CDKs and forming a complex. So what these cyclins do, mostly when they are released or when they are synthesized, they will bind to the CDK, that is the cyclin-dependent kinase enzymes, to form a complex, what we call the cyclin CDK complex. Okay. So what the complex will then do, it will now act as a signal to activate the cell to proceed to the next cycle. So I hope that makes sense. So let me repeat it. The third regulation mechanism is the use of cyclins. And cyclins are type of proteins that basically control the cell progression by activating cyclin-dependent enzymes, CDKs. So what the cyclin will do together with this CDK, they will form a complex. So the cyclin will go bind to the CDK to form a complex that we are calling the cyclin CDK complex. Now, this complex is where the main role is, because the complex will now be a signal, or it will be like an activator to signal the cell to be ready to transit to the next phase. So here we have a complex, and this complex is going to allow our cells at their different levels to be activated and move to the next stages. So we are being told that these cyclins, after they have completed their work, they will normally degrade, and then they will deactivate these CDKs, hence signaling exit from a particular phase. So maybe we are in G1 and you need the cell to move to S phase. So we'll have some cyclins there combining with CDK at that particular level to form a cyclin CDK complex that will signal the cell to move from one particular phase to the next. That is now from G1 to S phase. Once that transition from G1 to S phase, for instance, has taken place, then there'll be degrading and deactivation of these complexes so that we can exit the G1 and go to the S phase. So the cyclins are structurally related proteins whose levels, though, will be fluctuating throughout the cell cycle. So there are certain CDKs and cyclins that will be getting produced at the different levels. So different cell cycles events are regulated by distinct cyclin CDK complexes, and we are told we have more than 20 CDK related proteins and more than 11 cyclins that have been identified, and they play a major role in regulating our cell life cycle. So here is an example of these cyclins and CDKs. So starting with this first phase here where we have G1, S, G2, and M. So at each of these different cycles, we will be producing different cyclins and CDKs that will be combining to transition our cell to go to the next stages. So for example, at G1, we will find presence of cyclin D. We will also find CDK4 and 6. These ones will combine to give you a cyclin D CDK4/6 complex that will be activating our cells there and going through the processes. And then when we want to transit to S phase, we will have cyclin E's coming in. We have CDK2s, we have cyclin A2s. All these are different classes of cyclins and CDK complexes that are working together to allow the cell to be regulated and to transit from one cell cycle to the next. So here again, this is just an explanatory slide which gives more explanation. Like for example, what happens in G1? We are told in the G1 phase, this phase is initiated by cyclin D's, okay, binding and activation of CDK4/6 kinases. So we have cyclin D that is going to bind to CDK4 and 6 kinases, giving us a complex that we call cyclin D CDK4/6 complex that is going now to initiate, okay, the cell cycle at G1. And then when we want to transit to S phase, we are again being told that cyclin E, okay, will bind to CDK2 to form cyclin E CDK2 complex, and together with the initial complex, that is the cyclin D CDK4/6 complex, they will induce our cells to transit to the S phase. You see how it works? Very interesting. So that's how it happens. So we are told from G1 to S phase, we are having cyclin E binding to CDK2, working together with our cyclin D and CDK4/6 to induce the cell to transit from G1 to S phase. But again, in the S phase, it will do its normal work until everything is complete, and then we will need this cell to transit from S phase to G2 phase. So the transition from S phase to G2 phase, we will have other cyclins coming in here. So we have cyclin A2 combining with CDK2, transitioning the cell from S to G2. Going further, you see again that once we need the cell to transition from G2 to M phase, we will have cyclin A2B combining with CDK1, transitioning our cell all the way to M phase. And this is how the cycles work. But I need you to know that we are told the release of these cyclins and CDKs will vary at different stages. So in the next slide, I want us to see how the graph will change as we move from one stage to the next in regards to the cell life cycle. So this is how our cyclins will fluctuate as we move from one stage to the next. So remember when we said in G1 phase, we have cyclin Ds, and we move to the next phase, we have more cyclins coming in. So in G1, we start with the release of cyclin D. As we move to S phase, cyclin E comes in, more of cyclins A's, and then we finish into the mitotic phase with cyclin B's. So basically, this is how the concentration moves. So the cyclins get released and build up on each other as we progress from one cyclin level or from one phase to the next phase all the way to the final phase. So these are our cyclins: cyclin D, cyclin E, cyclin A, and cyclin B. And these proteins, like I said, will enable us to allow our cells to transition from one phase to the next by combining with what we call the CDKs, the cyclin-dependent kinase enzymes that will help us to activate and allow transition of the cell from one phase to the next. So thank you again for staying with us or with me to the last part of the class. And don't forget to share this with your friends, forums, or even places where people have similar interests with such content. Thank you so much and see you in our next class. Bye bye.